is that me? No, that's not me. Okay. From IIT Madras, one of the top institutes for engineering in the world. He then went on to do a PhD in applied physics from Stanford University and was the sole recipient of the 2001 American Physical Society PhD Thesis Award for beam physics. And then Shyam decided that that was not exciting enough, so he switched gears and he began investigating what DNA sequences, such as the non-coding conserved sequences, can tell us about gene expression and about human evolution. And he started doing that at Lawrence Berkeley National Laboratory with Eddie Rubin. He then joined GIS as an independent investigator in 2006, where his lab uses single-cell RNA-seq, cohort-scale ChIP-seq, and other high-throughput assays, together with algorithm development to uncover markers and mechanisms of human diseases. So his lab has many firsts to its name, including first single-cell transcriptomic analysis of colorectal cancer using the techniques I mentioned above. For more recently, Shyam's interests have expanded to applying these techniques to the development of the brain and its disorders, such as the idiopathic and syndromic autism. He currently serves as the Associate Director for Integrative Genomics and GIS Lead for the Center for Big Data and Integrative Genomics, a multi-institute consortium tasked with building the genome analytics infrastructure for Singapore's National Precision Medicine Program. So today, he's going to tell us about their recent work related to autism. And without further ado, I invite Shyam to the stage. Thank you.
Got it. Thank you very much, Ajay, for the very kind introduction. And thank you to Suresh and Ajay and Adam for bringing me here. It's a pleasure to connect with the neuro community, as always. And thank you, Jonathan, also for the very moving story. I think I have to drink a glass of water. That was very emotional. I'm not joking. So Histone Acetylome-wide association studies talk to you about genomic technologies and how they can help us learn something about disease, specifically autism and TB, and then one ongoing project recently that's incomplete on antidepressants. So for starters, what is autism? Autism, the definition has changed a little bit over the years. This is the latest definition. It's a combination of symptoms and impairment in social interactions and communication. That's on one side. And thank you. On the other side is repetitive behaviors and or you can say behavioral rigidity. And see, these are the two core categories of symptoms. But it's incomplete. There was some controversy in deciding the diagnostic criteria for autism because in reality, a lot of autists, perhaps most, if not all of them, if you actually look at it the right way, have sensory impairments as well. It's just that sensory impairments are not diagnostic criteria, but they have sensory impairments. So, and if you know anyone with autism, and I'm sure you all do, you might be familiar with that. So, and it's a spectrum. It's called autism. Great. It's called autism spectrum disorder because you have a whole range of severities. Okay, from some on the milder side, what used to be called Asperger's syndrome, all the way to on the more severe side, you have severe developmental disabilities and mutations in key genes. So autism is part of a syndrome. So you have syndromic autism where the individual may have many other symptoms, including these core deficits or these core impairments in autism, but they have other comorbidities as well, right? So it's a very broad spectrum, but they decided to diagnose it, create the diagnostic criteria as though it's one thing, right? Because it was hard to draw a line. And that's a key thing to remember. And the causation is complex, extremely complex, I would say. So it's caused by genes plus environment. People who, let's say, I'll talk about the environmental causation. I think you understand what genetic causation means. You have a mutation in the genes that contributes to the risk of autism. Environmental causation is less well appreciated, I would say. So toxins or maternal infection during gestation is a strong risk factor. For example, if the mother worked with chemicals, let's say worked in gardening during pregnancy, that dramatically increases the risk of autism for the child. So that's an example of toxins, exposure to toxins prenatally. So it's a developmental disorder, okay, and very, very complex causation. There's no one autism gene or one autism cause. And because of that, there's no, you can say, cure or no, there isn't even a drug for autism, okay? So people with autism get ADHD drugs, but ADHD is not a core symptom of autism. There are no drugs existing right now for the core symptoms of autism. What you have is cognitive behavioral therapy and other non-drug therapies, right? So it is a really, and then I'll tell you the other important fact, which is it's, in the US, the latest study is 1 in 60 kids at age 8 has autism, right? So that's a staggeringly high number. And some people say, based on self-reports, this number can be even higher. People have reported numbers of 1 in 40 kids in the US. Of course, there's a whole range of numbers. There's other older studies saying 1 in 400, 1 in 500. A big part of it is simply that autism is a spectrum, okay? So if you cut the spectrum here and say, I'll diagnose only these people, then your measured autism incidence will be lower. If you cut the spectrum here and you say, I'll diagnose these people, then 1 in 59 will have autism, 1 in 40 will have autism. To some extent, it's subjective, okay? But then lots of disease diagnoses are subjective. You have to draw a line somewhere, and certainly people on this side of the line, they have something in common, right? At least they have something in common that the clinician can see, okay? They have these symptoms. But do they have something in common over here? At the level of genes, at the level of environment? That's what we really want to know, because we want to change the situation that there's no drugs for autism, right? And traditionally you say, okay, find the autism gene, or find the disease gene, find out what are the consequences of that gene being mutated, and then design a drug for that gene. But in the case of autism, it's estimated there's 200 plus genes. So what are you going to do, right? You cannot design a drug for every single one of those 200 genes. In fact, the drug company will just refuse to do it because it's too much money invested for a narrow slice. So autism as a whole is common, but if you divide it by prevalence by 200, it's a rare disease, right? Every specific genetic form of autism is a rare disease, and many of these genes contribute very slightly to autism. It's some kind of combination of all of these. So that is really the challenge we're facing. No drugs, very little understanding of what's shared. We know that they share the diagnostic criteria. They do not share the genetic traits. So somewhere between the genes and their behavior, they converge, right? So where is it? At what level is that? So you have the genes, and the genes influence the molecules in the cell, their abundance and their properties. Molecules in the cell influence how the neurons, for example, behave. The behavior of the neurons influences the circuits, the behavior of the circuits in our brain, and the behavior of the circuits in our brain are observed as our behavior, right? So you've got that hierarchy of effect from the genes all the way to the observed behavior. At the level of observed behavior, it's one thing, you can say, or at least one spectrum. At the level of genes, very different. Does it converge immediately and become one thing just above the level of the genes? Or does it converge very late? We don't really know the answer, okay? But I'll give what we believe is the answer, right? I was just at a conference in New York where people just stated, one of the presenters just stated that as a fact, that the convergence happens only at the level of neural circuits. Because he was aware that there isn't much convergence at the level of genes, but there are layers in between, and those are the layers that we're going to look at. And I'll already tell you the answer. We found convergence much below the level of circuits, right, at the molecular level, and just about the level of genes. So also I said, it's very common. I got this picture off the internet, actually. I don't know this person, Kapi Rajalingam, but I know lots of people like him who have autism, and I'm sure you all do. So that's what motivated the study. So I'm a physicist originally by training, then moved into genomics. Never imagined in my wildest dreams that I would ever do anything that could actually make a difference in the lives of patients, right? I thought I'd just keep on doing mathematical stuff. But maybe, hopefully, you'll be convinced that even a mathematical person like me is starting to move in the direction of this, and that the field itself, is not me, the field itself is really, really exciting, using big data and mathematical analysis to try to find patterns where traditional biological methods don't see any commonality and they don't see any patterns. So genetic basis of autism, what do people do? They take DNA from lots of patients, and they take DNA from lots of non-patients, sequence that DNA, and say, are there any systematic differences? Okay, is this mutation present more in patients than in non-patients? Or is that mutation present more in non-patients than in patients? If so, then that mutation is, has something to do, is potentially causal for autism. And as I said, there's 200 plus mutations like that. Not all discovered by this method, but quite a few. So people said, okay, that's not very satisfactory. Let's look at molecules in a slightly different way. So then another approach is to say, let's not look at the DNA sequence itself. Let's look at what's on top of the DNA sequence. And that's not really the definition of epigenetics, but you can imagine that it is the definition of epigenetics. It does make a huge difference. So what's on top of the DNA is all these chemical groups, all these chemical modifications on it that affects how the DNA expresses itself, okay? Because the DNA is wrapped up, the cell has to read the DNA and interpret it, and then get its instructions from the DNA. And so these chemical modifications, this is one possible chemical modification, a very common one. It affects how the DNA expresses itself, and it's not directly related to the DNA sequence. So you can look at what's called an epigenome-wide association study. Look at the methylation groups on DNA in patients versus control. Look at differential methylation, and then try to see if you find something, let's say in autism, that's specific to patients or specific to controls. Now, this is also a relatively well-established field. And we said, and I would say the correlation between these traits and other molecular traits was not as strong as people would have liked. And so we said, let's try something else. Okay, let's look up just above the DNA, and let's look at chemical modifications, but not these chemical modifications. Let's look at something slightly different. So before I come to that, I'll just say this epigenome-wide association study paradigm is challenging. It's not straightforward. But as is often the case in life, those challenges are also advantages, opportunities if you look at it differently. So if you're looking at DNA, right, the DNA in your blood and the DNA in your brain is more or less the same. So if I want to see what mutations are affecting your brain, I can just sequence the DNA in your blood. But the chemical changes on top of the DNA are different in different parts of your body. So you want to try to profile the chemical changes in the brain. Blood is not good enough. And then access, obviously, to brain tissue is much harder, right? But at the same time, if you do get access to brain tissue, then you get much more specific information relevant for autism, right? So that's the advantage of using this approach. And then the disadvantage is that, a genetic mutation, if you find it increased in, say, autists, you'll say autism could not have caused that mutation. It must be the mutation causing the autism, right? So the causality is very clear. Whereas if you look at a chemical change on top of the DNA, epigenetic change, it could be an environmental influence. It could be caused by the autism itself rather than causing the autism. And it's quite complex. But at the same time, as I mentioned, autism is caused by genes plus environment. And so we really want to go above the DNA and look at molecular traits or molecular features that can tell you something about the other non-genetic influences as well. because they are affecting the behavior of the cells and the behavior of the neurons, the behavior of the circuits, and the behavior of the person. And I mentioned this cause or consequencing. So epigenetic features are consequences as well as causes, right? And then, as I said, it's just DNA methylation. There are other epigenetic marks you can look at. Specifically, right, histone modifications. What are histones? Histones are these kind of spools that the DNA thread is wound around in the nucleus, so that it can be very, very compact. If you stretch out the DNA, it's huge. So if you wander around the spools, it's really tiny, and you can fit it into the nucleus, and it doesn't get knotted, and so on. So now these spools also, right, they have these proteins bunched together and form the spools. They have these tails sticking out of them, and these tails normally stick to DNA because DNA is negatively charged and the tails are positively charged. But then you can have chemical modifications of the tails so that now the positive charge is cancelled on the tails, right? You add a negative group. So then the tails are repelled or don't stick to the DNA as strongly, and then the DNA is able to express itself more easily, right? Because it's not tightly wound around the spool. So that's called open chromatin. Okay, so if you have certain chemical modifications on these spools, on the histone proteins, it releases its tight grip on the DNA loosens up, and then the cell can see that piece of DNA and read it out, right? So clearly, even if there's no mutation there, simply adding these chemical changes will change the behavior of the cell. So this is really what we wanted to look at. Do these chemical changes in the packaging of DNA, do they have anything to do with autism? Okay. And there are many possible chemical changes in the packaging, as I show here, but there's really one that's kind of that rules them all in some sense in the field. And that's histone H3, lysine 27 acetylation. So it's histone H3, this is lysine 27. So here we're showing methylation. So, but instead of methylation, it can be acetylated as well. You add an acetyl group to it, like these groups over here, okay? And if you do that, then this lysine, which is normally positively charged, it loses that positive charge, and then there's some slight repulsion. And we and others have shown that this mark, this chemical mark, is a signature of the switches that control gene expression. And those switches are called enhancers and promoters. So when you have this chemical modification, that particular DNA sequence acts as a switch. And it acts as a switch that turns the gene on and allows the gene to produce RNA and then proteins, right? So it's very nice to say, okay, now if we just read the on-off state of all the switches and say which switches are on in the brain, which switches are off, and is that different in autism versus control, right? Then that can tell you something about the behavior. And that's another example of looking just above the DNA to see, is there anything that's common? It's still a molecular thing we're looking at, but it's not exactly at the DNA level. No one had looked at it before when we started this. No one had looked at it before in autism or really systematically in any disease. No one had done disease versus control studies systematically looking at any of these acetylation marks when we started. And there was one study of moderate size on methylation marks. Excuse me, for any disease, which we thought was crazy, right? But why would that be? So we said, let's take that on. Let's do a histone acetylome-wide association study, look at these acetylation changes in patients versus controls. And we said, let's go after autism for all the reasons I mentioned before. So there was a collaboration with Dan Geschwin, UCLA, is a well-known autism molecular biologist, and John Mill at Exeter, who was a pioneer in epigenetics. And we were kind of newbies and newcomers to the field, but we knew data analysis and we knew how to profile this histone modification. And so the study was driven by Wenjie and Jeremy in my group. So this is already published, it's old stuff, but for people who have not read the paper, which is presumably the majority of you, I guess it's still new. So what did we do? We took post-mortem brains, a few different brain regions, prefrontal cortex and temporal cortex, which are thought to be, so prefrontal cortex is executive function, and temporal cortex is associated with speech and language. So both of which are impaired or altered in autism. So these are plausible brain regions for looking at chromatin packaging or DNA packaging in autism. Okay. And then we took cerebellum as a negative control. There's some studies saying that there are changes in cerebellum also in autism, but there's much less known about that and much less evidence, I would say, about the involvement of cerebellum. So we took about 40 versus 40 case and 40 control post-mortem brain samples from this brain region, from temporal cortex 30 versus 30, from cerebellum 30 versus 30, and used this whole genome high throughput molecular technique to read out this chemical modification throughout all of the DNA in these chunks of tissue. Okay, at every location in the genome, we looked at this. And then, the genes are strung out along the chromosomes, and we wanted to say, if we find some changes in the chemical modifications, at which positions in the chromosome do we find them? Which are the genes nearby? And maybe that can tell us something about what's going on in autism. So the first thing, now remember that if you do this instead of chemical modifications, you do this with DNA, you'll find nothing, okay? You have to do, the signals are so weak, you have to do like 20,000 autism versus 40,000 control to see even the glimmer of a statistical difference between autism and control if you're looking at DNA sequence. Now this is audacious, right? We're doing 30 versus 30, or 40 versus 40, so much smaller study. So what we are presuming is that the statistical signal, the difference we're looking for is huge. And if it's a huge difference, you can get away with a small sample size. You'll see it even with a small number of individuals. People told us this is just crazy and would not work, except, of course, Dan was quite sure it was working. He was our collaborator. And as it turned out, we found 5,000 locations in the genome that were significantly different in both brain regions, right, between autism and control, and they're highly correlated. So this was dramatic. So this told us that even though at the DNA level, autism is many, many things, at the level, just one layer above that, at the level of the packaging of DNA, autism is to some approximation one thing, right? And most individuals with autism, if you look at their postmortem brain tissue, they will have these 5,000, they will have changes in the chemical modification at these 5,000 loci. That's remarkable, right? So there is a commonality at the molecular level. It's not just at the circuit level. Now, what do I mean by differences? So this is an example of what we actually look at in the data. So here what we're showing is one segment of the genome, one segment of the DNA in our cells. And these are 3 individuals in red. So these are 3 brain samples from autism, and these are 3 brain samples from control, and this is one gene. You can see here, and then what this bump says is that you see a lot of that histone acetylation, histone chemical modification in this part of the DNA in autistic individuals. But you can see the bump is kind of flat here. You don't see that change in control individuals. So this is the kind of thing we're trying to find. Something that's shared across all autists and not there in controlled individuals, or vice versa. Sometimes something that's shared across all controlled individuals, but missing in individuals in brains from people with ASD. Right? And we found 5,000 such locations in the genome. It's quite dramatic. Now, are there any common themes? So this is one gene, right? And this 5,000 regions, maybe near 2,000 genes. What are the common themes? We found some common themes. Synaptic transmission, everybody already knows that synaptic transmission, the connections, the signaling between neurons is altered in autism. Okay, so this was, you can say, a so-called positive control. If we did the study correctly, we would find this, and indeed we found it. Epilepsy also, a lot of individuals with ASD have epilepsy, so this is not surprising. But then there was some funny stuff that's more exotic, okay? So zinc ion homeostasis, histone deacetylation activity, and so on, and some immune pathways that seem to be coherently changed at the level of DNA packaging. And then when we read about them, we started to find papers, okay, even zinc ion homeostasis. There's 1 or 2 papers saying that low zinc is a predisposition for autism. It's not a widely held view, and the study size is small. So, people won't believe that zinc is a cause, malnutrition in zinc is a cause of autism. But our data does seem to suggest that these zinc genes stand out in terms of their differential activity in the ASD brain. So this is an example of when you do this massive data generation exercise, you see some of the expected usual suspects, so you don't learn a lot there, and then you see these intriguing new signals that are worth following up, that no one would really have paid attention to from that individual paper that someone published out of the 1,000 papers on autism that people have published, right? But this is unbiased. We were not specifically looking for zinc ion regulation, and it just popped out, and so we believe it more. Now, more broadly, I talked about having shared signals across all individuals with autism, okay? Now, here's a dramatic form, a severe form of autism, where an entire arm of a chromosome is duplicated. It's not just one small change. Now, we had 9 individuals with this severe syndromic form of autism. We compared their changes on the x-axis to the changes in the idiopathic autism individuals, the individuals who had autism for no known reason, okay, and generally milder forms of autism. You can see here that there's highly correlated. Each dot is one location in the genome. Now, at all locations in the genome, the changes we saw in these mild autism individuals were highly correlated with the changes we saw in the severe autism individuals. So that's, again, a real surprise, right? Because if you look at their symptoms, so individuals with this, they actually have, their body structure looks different. It's not just that they have autism. So you'll say, wow, there's no way at the molecular level they can be the same. Sure, you're calling it autism because you have some clinicians came up with some arbitrary set of criteria, and so these people are called autism spectrum disorder, and these people are called autism spectrum disorder, but look at them, they're really not the same, right? But what we're finding is that they are the same at the molecular level, and if you look closely, this line is tilted slightly down, okay? So whatever changes you have in people with a milder form of autism, you have a slightly stronger version of that chemical change in the individuals with a syndromic severe form of autism, which again makes sense. So it says, again, this goes with the spectrum concept, right? So even if these people with a very severe form of autism are on the same spectrum, if you define the spectrum on the basis of severity of these chemical changes in the DNA. So here's another example of rare forms of autism that are syndromic having a connection with chemical changes in common forms of autism. So HDAC4 is a gene. When it's mutated, it causes what's called brachydactyly mental retardation syndrome. And if I remember correctly, that's a split hand syndrome with mental retardation. And it's a syndromic form of autism, so it also includes autism symptoms, right? So people with this syndrome have this gene mutated, but people who don't have this syndrome in the milder form of autism, they have these chemical changes at all these locations in the gene locus, and the chemical changes are in such a direction that they reduce the activity of this gene, right? So that's the molecular severity scale. If you kill this gene altogether, you get the severe form of autism and other symptoms as well. If you just mildly push down its activity, then that contributes to the milder forms of autism, right? So the molecular severity correlates with the symptom severity. And so because it's a rare autism gene, no one would ever thought to design a drug to it. No drug company would go after it because it's so rare they won't make any money, right? But now what our data is showing is that this gene is relevant not just for these individuals. It's relevant in all individuals in autism, right? Now suddenly it becomes a potential drug target, right? I'm not saying this specific gene should be pursued as a drug target, but this is an example of how when you connect rare autism to common autism, then, and at the same molecular loci, then you start opening up possibilities for drug development. So another thing we did with the same data set, right, and again, my training is on the more mathematical and engineering side, we realized that we can take our data, which is, you use some DNA sequencing to read out these chemical changes. You can take our data and say, okay, at each enhancer promoter, at each switch region, we can read the DNA and say, okay, some individuals have a G or G nucleotide, DNA is ATGC, some individuals have a G here, Some individuals have a G and a C, and some individuals have a CC. Right? So these are just different variants of this one mutation in different individuals. We can actually read that out, and we can correlate it. So we can say individuals with a CC mutation, they have very little histone acetylation here, so that gene is, the switch is kind of off. And individuals with a GG mutation, they have a lot of histone acetylation, so the switch is more on. And so we correlated the mutations with the onness of the switches. And so then we said, if the mutation has some correlation with how on the switch is across the whole population, it must be causing this switch to turn on or turn off. Right? So then we can connect our genetics to the chemical modifications, the epigenetics. And so everything that people know genetically about these diseases, we can start to see how does it connect to the packaging and behavior of the cell. So we came up with a statistical test for that, and we found lots of interesting loci. So this is a mutation that is associated with risk of a shared risk across 5 different psych disorders, including autism. And it's in a calcium channel gene locus. And you can see here that individuals with CC have less histone acetylation here. Individuals with GG mutation have more histone acetylation here. And so this gene is presumably more active in these individuals than in these individuals because this switch, the activity of the switch is affected by this mutation. So this is an example of where you take mutations that are already known to have some effect on disease, and you can start to say, why does it have an effect on disease? Perhaps because it affects this switch, which then affects the activity of the gene. Right? So that's a nice side effect of the data we generated. And this is always a paradigm, I would say, in genomics. When you generate a massive data set of this complexity, there's really no limit to the number of ways in which you can analyze it, the number of lenses through which you can look at it, and the number of conclusions you can draw from it. I don't think we've drawn all the insights from this data set that you could draw. In fact, there's one more result I'm not showing here. We debated whether, again, some kind of statistical analysis of this data. We debated whether to put it in the manuscript because it seemed it is just pure statistics. Okay, we had no follow-up to say, are those statistical predictions really correct? So we said, well, at least if someone has published a paper saying those predictions are correct, then we'll be reassured that we're doing the analysis right. Okay, no one had published anything about that gene, and the reason I'm being very vague about this is that recently, at that conference in New York, I met someone who's a clinician who sequenced the DNA of lots of individuals with autism, and he said, aha, I want to talk to you, because that statistical finding you had in that paper of yours, we just found that that gene is mutated in autism. And it's a completely new gene that is not known to be an autism gene. And it has, it's amazing that we found it at the DNA mutation level, and you found it at the gene activity level, right? At the DNA packaging level. So he wants to work with us on this. So just a long story short, massive omics data sets are a kind of non-traditional way to study biology, but is the only way physicists like me know how to study biology. And you can't go in saying, I want to find this. So it's not hypothesis-driven science, so to speak. But you can actually find stuff you didn't expect to find. You can find many such things like that, and that's the power. So to summarize our autism work, this was the paradigm, okay? So you can think of a funnel. You have mutations in genes. You have mutations near genes that are causing autism. You have structural copy number variants, so to speak, where this, like Dup15q, this entire arm of a chromosome has extra copies or fewer copies. All of that is known to cause autism, environmental toxins, maternal infection during pregnancy, and all that. Very, very diverse. You cannot design a drug to attack the causation over here because it's so diverse, right? So what our results suggest is that this funnel narrows out very quickly just above the DNA, the level of DNA packaging. And so if the coherence that you see in the symptoms is mirrored by the coherence you see at the chromatin. So at the level of chromatin, autism or packaging, autism is one thing. I'm oversimplifying. It's this variability, but it's to some extent true. So if you want to design a drug, don't necessarily attack the causal mutation. Attack the downstream causes of that causal mutation that seem to be quite common regardless of where the mutation happened. Now, this is a paradigm we came up with looking at our autism data, but now when I travel around going to conferences, people say, oh, we independently discovered this paradigm in our own study using our own methodology on our own disease. Right? So I don't think it's specific to autism. It's quite common that even when you see genetic diversity and almost chaos, you see that at the level of packaging and chemical changes of the DNA, it's coherent. And so that's potentially where you can attack things with drugs. And also study things with read things out with diagnostics. So after we published, so that was the first Histone Acetylome association study. Then our collaborator John Mill fairly soon published a similar study using histone acetylation on Alzheimer's disease, similarly small cohort size. So again, proving that you don't need 20,000 individuals. That's a real power of this methodology. A few dozens of individuals is still enough, right? And then after that, another group published, I think this was a few hundred. So they went for the traditional paradigm of you need lots of individuals. But John Mill got similar signals just from a handful of individuals, so 20 versus 20. And then we're taking this forward. We're studying TB now with this, and there's a neuroscience symposium, so I won't talk too much about TB, except to say that the methodology is quite general and can be applied to any disease. And as a physicist, that's what I like. I'm a methods person. I only collaborate with disease specialists to apply those methods to specific cases. So TB, just quickly, I'll give you the punchline. Okay, what's different in our TB study is, even though we felt our results on autism were very exciting, there were still some skeptics, and justifiably, saying that, well, you say you found these differences between autism and control. And you say that you found the same differences in the prefrontal cortex and the temporal cortex, more or less. And sure, you found some differences that are in known autism genes, but I'm still not convinced that those differences are real. They're not technical artifacts. So really the acid test and things like this is you do the study on this group of people, find some differences, then do the study on that group of people, find some differences, and then you correlate. Right? And you say, are they reproducible? So so with the TB study, we did the study design like that. So we had a discovery cohort, and we found some histone packaging, DNA packaging differences. Each dot is one location in the genome where we saw a difference. We had a separate validation cohort where independently we generated data, and we said those same dots, those same parts of the genome, do they look different between TB and control blood in that second group of individuals? Now, if you just look at the black dots are the genomic regions that we said are significantly different. And you can see here that the significantly different parts of the genome in the discovery cohort and the validation cohort, they're different in the same direction. If it's up in the discovery cohort, it's up in the validation cohort. If it's down in the discovery cohort, it's down in the validation cohort, right? And the same for these two different cell types. So this is really the first time anyone has validated this methodology in an independent cohort and shown that it's robust and reproducible, despite the fact that we had only 20 versus 20 individuals in our discovery cohort, and I think 14 versus 14 individuals in our validation cohort. So it's an astonishingly small number of individuals, number of samples. That means that I don't need to collect a consortium of 10 clinicians in Boston or New York City or some big academic medical center and get 10,000 individual samples to do the study. One scientist, so like myself, say a genomics person, collaborating with one clinician in a small country, perhaps like Singapore. So the TB study was from Singapore samples, can collect enough samples to learn some biology and get some reproducible signals. And so that really democratizes this kind of disease versus control association study, if you can do it in such small cases. And I can't make this point strongly enough because the wider community still doesn't believe it. There's been only a handful of histone acetylation association studies in the world. There are more than 1,000 genetic association studies in the world. So if you talk to anyone who's an expert on association studies, they'll say, it's impossible. You cannot do this study with 20 versus 20. You say, here's my data, and they say, no, it's impossible, you cannot do it. Yeah, I've proved it, it's reproducible. No, it's not possible, you cannot do it. So I keep beating this drum. Eventually enough people will publish studies like this, and then people accept that the cohort size you need for DNA mutations is large, but the cohort size you need for this kind of change is quite small. And so this is a very valuable adjunct to DNA-based, mutation-based studies. And so in this case, we didn't just stop at genomic signatures. We actually found an ion channel gene, okay? And I won't explain in detail what all these things mean. But let me cut to the chase, okay? So we found a gene that's an ion channel that's different between TB blood and control blood. Okay, so if you have TB infection, your blood cells have more of this ion channel. So that means it presumably allows more of potassium to enter your cells. And it turns out that if we increase potassium, we simply increase extracellular potassium, and here what we're showing is on the y-axis, how many infected cells do you get? You culture these immune cells, infect them, and you see how well the infection, how successful the infection is. If with control, okay, the infection is this successful, you get so many infecting microbes. As you increase the ionic concentration of potassium, the infection level goes down in these cultured cells, right? And then we did many, many other experiments to show that this gene that pulls the potassium ions into the cell is contributing to apoptosis and other mechanisms, other defense mechanisms the cell has against the pathogen. And so that's why when you get TB, these cells ramp up the level of this ion channel to protect, and that it protects them. They're infected, of course, so they did not successfully fight the infection, but they're trying to fight it, right? And so when you give them TB drugs, it's not just a drug that's curing the patient. It's the drugs plus the patient's own cells themselves that are pushing the infection down. So this is one example. And then the thing about TB is you have multi-drug resistant TB now. TB, actually I should have mentioned, is the biggest killer of all infectious disease in the world. It's bigger than HIV/AIDS, right? So, and multi-drug resistant TB is on the rise. So it's quite a scary disease, the most scary infectious disease, I would say. And you want new drugs, you want new pathways, you want new understanding, new players in the response to TB. So potentially, you can improve the efficacy or improve the activity of genes like this potassium channel, and that can help the body fight the TB. And so you can have new TB drugs that hopefully these bugs won't be resistant to. So to summarize, I won't belabor these points, I've said them already. And so everything I've shown you so far is epigenomics or histone acetylation profiling the packaging of DNA in so-called bulk samples. So bulk samples is you take a lump of tissue or say 5 ml of blood and extract the cells. You've got millions of cells there, hundreds of thousands of cells. You mash up the cells, extract the molecules, and you count them, right? So that's a bulk sample. Now, the obvious problem with that is that actually you want to look at it one cell at a time, right? Because these different cells in your tissue sample all have different properties. So that's what we do here at the GWAS Single Cell Omics Center. We have multiple technologies to do that, to essentially turn a tissue into a bunch of dissociated individual cells and then count molecules in each individual cell using these technologies. And this is a platform that's open to all of Singapore, so anyone who's interested in single cell analysis is welcome to use this. Just email me or come to the GWAS website. You can email Sudhagar, who runs the platform, and we'll provide access to it and help you generate the data. And these are some of our customers, so to speak, and collaborators. So I'll skip this because this is a very preliminary study, except to say Michael Meany at 6 and Jan-Paul Buschdorf also at 6 are our collaborators on this, and Nirmala Ryan is driving this project with support from Vibhor Kumar, who's a computational biologist. Here we're trying to say, just like we studied autism using his chemical changes to histones, we're trying to say, what does Prozac do to your brain? Right? What is your brain on Prozac? It's one of the most widely prescribed drugs in the world. And it's very complex. It's a serotonin reuptake inhibitor, so the name makes it sound like we know what Prozac does in your brain, but we don't. Right? So Prozac affects your whole brain. It doesn't just affect serotonin reuptake, and it has severe side effects. But most of the literature, molecular literature on Prozac is just specific brain regions, specific cell types, specific molecules. Right? So the beauty of omics is you can say, I won't be hypothesis driven. I won't predict where the interesting information is going to lie. I'm just going to generate a massive data set. So we said 27 brain regions. We drug-treated rats, Prozac-treated rats, 27 brain regions, drug-treated versus untreated, single-cell analysis, look at all cell types, one cell at a time, and see which cell type is really giving us the signals. So this is early days. I'll say, we're still analyzing the data, but we're seeing some interesting stuff over here. This is what the data looks like, okay, single cell data. Each dot is a cell here, and cells that are near each other are the same cell type. So you can see microglia, so they're the immune cells of the brain. They're all near each other, but they don't look exactly like each other in terms of their gene expression. So these are astrocytes, so I think these are astrocytes here. And then or maybe these are astrocytes, I don't know, oligodendrocyte progenitor cells. And so these are all oligodendrocytes. So different cell groups separate out, and then even within the same major cell type, neuroscientists will say, okay, this is subtype A, this is subtype B, this is subtype C, and so on. But when you actually look at the gene expression data, RNA data, they look like they're on a continuum very often. So they're discrete islands, and within one island, the different subtypes of cells are on a continuum. So what we want to say is, at each location in this continuum, what do the drug-treated cells look like, and what do the control cells look like? And so we can say, okay, these cells are responding to the drug. All right, these cells are not responding to the drug. These cells are responding very strongly to the drug, and so on. I can take a hint. And that's all. I'll skip the rest. And single cell analysis is exciting. It can help us understand the complexity of cell types in a tissue, one cell at a time, okay? And the best way to do it actually is using imaging rather than dissociating the cells from the tissue. What we're doing right now is, so each dot is one gene, the RNA from one gene. Each color is RNA from one gene, and this is a tissue. So now we're not bursting the tissue apart and looking one cell at a time. We're imaging the molecules in the tissue environment, and that's a much more powerful way of looking at it, because then we can see actual structures. This is even more early days. It's a very difficult technology. Just setting it up. It's a massive team that's setting it up, and I'll show a photo of the team. And the data analysis is also challenging, to see patterns in these molecular pictures. And right now, the molecular patterns we see look kind of silly, but we'll see more accurate patterns soon, I think, once we refine the analysis. That's the spatial omics team. And I should mention my collaborators as well. I should thank the NIH, Psych and Code, and A* for funding all of this. That's the team. And lastly, if you're interested in single-cell research, we're organizing a conference in Singapore at the GIS, in the Biopolis, actually, just a month and a half from now. So please come by. Registration is free. We have all the top single-cell researchers in Asia, all the leaders of all the single-cell in all the major Asian countries and Australia coming and speaking at this event, and we have some speakers from outside Asia as well. And we really welcome more and more people. The idea of this meeting is to bring people into the single-cell field and contribute Asian samples and Asian data to international consortia. Thank you.
Thank you, Shyam, for giving us a glimpse into all the different diseases you guys are approaching with these techniques. So there's some microphones on the back, and yes, Jongwei.
Thank you, Shyam, for the fascinating talk. I want to ask you about the autism study. So let's say if you chose to use mRNA instead of the acetylome, would you predict that you will need bigger or smaller cohort size to see differences?
Great question. Actually, Dan Geschwin did the RNA study on the same brain samples. And so his paper was published like 1 month after our paper. Same cohort size, obviously. The signals he found were weaker. We don't think it's because the RNA signals themselves are weaker. I mean, his p-values are lower and so on. It's mainly that in postmortem samples, the RNA degrades very fast. So whatever was there initially, you see a very distorted version of it. And that's why looking at histone modification is better, because those things don't change so fast in postmortem samples. But RNA is also very, very useful.
Yeah, Shyam, thank you for the great talk. I guess my question is a bit more broad. So you found all these acetylome signals, and you see that, do you think they're causative or correlative? Do you think they're causing tuberculosis or autism, or they're actually a sign of something else?
Great question. So as I alluded to in one of the very early slides, they could be cause or consequence. All we know is they're associated, right? There's correlation. Some of that correlation is causation, some of it is not. Where do we go from there? We look at the literature. So we say, is there something plausible? Okay, I'm seeing zinc-ion homeostasis. There's some hints of it in the literature. I have these 4 regions that seem to be different in zinc-ion genes. Let's pursue those. There's a chance they'll be causative. HDAC4, we already know that the gene mutation is causative, right? So then the histone acetylation changes are likely also causative, but milder. So where other people created a genetic mutation in a mouse and studied the mouse with no HDAC4, we can study the mouse with lower histone acetylation and see if that gives you milder symptoms. So those are some of the avenues you can pursue. Or you can do something crazy and say, like in TB, we said, look, this ion channel gene, massive signal statistically, nothing known about it in TB, but it's the biggest signal we're seeing in monocytes. So sometimes you have to take a leap of faith also, and there's no literature on that, right? And at least in my career in genomics, and I've been doing genomics for 16 years, if you do the statistics right and you do the experiment right, your top signals in the genome, the ones that you know nothing about, they turn out, it is my anecdotal experience, they turn out to be very, very relevant for mechanistically, and they turn out to be causal. Like that ion channel gene turned out to be causal. So if you think about, if I can rationalize it, that's the empirical observation. How do you rationalize it? You say, well, why is the cell creating such a strong change if it's irrelevant, right? So probably relevant.
Hi, that was a fascinating talk. I'm wondering then if the conclusion from your talk is that there's no clear genetic mutation that on its own causes the syndrome, and that this is a complex genetic and environmental type disease. So is the conclusion that perhaps something in the environment is the main trigger of autism, and that we still don't know what that might be, but nothing genetic per se is the trigger, that it's perhaps an environmental trigger that creates these epigenetic mutations that you're seeing?
Okay, so I wouldn't say there's no genetic cause. So if you look at twin studies, so different people do twin studies differently, and they estimate that something between 40 to 80% of autism causation is genetic. Right? If you look at identical twins versus fraternal twins. So it's strongly genetic, and the rest of it is presumably environmental. So what I was trying to say was that the genetic mutations are very, very diverse, and mostly mutations of small effects. Not that the mutations are not causing autism, it's just that it's a huge combination of mutations of small effect that are causing autism, and a lot of the histone epigenetic changes we see, they're probably downstream of the genetic changes. In this person, you had these 3 mutations. In that person, you had these 5 mutations. They all cause the same epigenetic change. So that's one route.
The question is why? Why do they all cause those 5,000 epigenetic peaks? Why?
Let me come to that. I also want to say it's not just the mutations that cause those 5,000 changes. It's also, like you said, environmental influences as well. Why those 5,000? Why is that shared between all individuals? My guess is that because how are those, not 5,000 individuals, 5,000 loci, how are those 40 individuals chosen for our omics study? They're chosen because they had coherent symptoms, right? So what our data are saying is that if you have those symptoms, it's probably you have those changes in histone acetylation. That's how those symptoms arose. To have those symptoms, you have to have these changes in these regions, right? That's one direction. The other direction is the other way. The symptoms are causing those epigenetic changes. So it's both. Like I just make a number. To have those symptoms, you need 400 of those 5,000 loci altered. And once you have those symptoms, the remaining 4,600 of those loci are altered. And since all those individuals with autism had autism, it would cause all those 4,600 loci to be changed in that manner. Right? So it's some combination of being downstream of autism and being necessary for autism.
Question there, please.
Hello. I was curious why more emphasis wasn't being put on the HDAC result. Does the histone deacetylation or the deacetylase, the state of that gene, does that not explain the acetylation states of all the other ones? If the HDAC gene affects the acetylation state of everything.
Why is there not enough emphasis on that gene?
Yeah, is that not a causation?
Okay, so no, it's thought to cause autism. It's just that it's thought to cause a very rare form of autism. Okay. So now our data suggests that even in common idiopathic milder forms of autism, suppression of that gene may be playing a role.
Yeah, I was just meaning, so you said as the gene has more and more markers, the level of autism increases.
Oh, more and more severe mutations, you can. So more and more severe alterations.
Yeah, so does that, but the fact that this gene controls the acetylation states of everything in the DNA, does that not explain the acetylation results of everything else?
Excellent
if it
question.
Can remove
Excellent
acetylation
question.
marks.
Okay, so I don't think any of our individuals, maybe at most one, had that gene mutated.
Oh, okay.
Right? So if there's histone acetylation changes near that gene, it's because of something else, some other mutation, or because of some environmental perturbation. Mm-hmm. That gene was non-mutated in these individuals.
Okay, but was it acetylated, changed? I thought your slide showed that the HDAC gene was the most correlated.
The acetylation was changed.
Yeah.
For some other reason, right? Not because of mutation in that gene. Mm-hmm. But I think what you're getting at is now that the acetylation of that gene has changed, for some external reason, that gene has lower expression, and that is going to, in turn, change acetylation globally in the genome. Quite likely.
Yeah.
Quite likely.
Yeah. Okay. So that was.
So I guess a follow-up question to that would be, do you see similarity between HDAC4 knockouts and the things that you're looking at, right? So that would point to HDAC4 as a common pathway of those two things, and it sort of feeds into. Have you looked at the HDAC4 mutants or?
So this is the only data set we have so far, and we had no HDAC4 mutations in our cohort. The only severe syndromic mutation we had was a dup15q.
What was a
duplication of 15q, chromosome 15q. We don't know, we don't have data on the epigenetic effects of HDAC4 mutation.
Right.
But yeah, it's an excellent question, and I take your point that can in turn cause global chromatin changes.
Yeah, I guess what I'm saying is the epigenetic state of the HDAC itself could then go on to influence everything else.
Yes.
So I see that there are a lot of questions, but maybe we need to take a call about bringing this outside. Shyam is here for dinner. So what I wanted to say is, first of all, let's thank both the speakers. Shyam, for this wonderful talk. All right, one question. Let's go with one question. One question.
All right, great talk. My question is about that you said that you took the chunks of brain from autism patients and control patients, right? What are those brain regions? And as a background in neurosurgery, I'm just wondering, are those brain regions might be connected? And can we just narrow down to omics like cellular, are these astrocytes or neurons? So where are the changes? And then are these brain regions somehow connected? So we can narrow down.
Certainly. I'll give you the dumb non-neuroscientist answer. All brain regions are connected. Beyond that, don't ask me for specifics. But certainly, in the prefrontal cortex and temporal cortex, the chunks, the lumps of tissue we profile, we just mash them up, so it's bulk analysis. It's a mixture of cell types. So that's why we're excited about single-cell analysis now. We just got funded by the Simons Foundation Autism Research Institute to do single-cell analysis of fetal brain samples to see how the brain develops, what states you see there. And then we're doing single-cell analysis of autism versus control samples to see exactly in which cell types the differences manifest. Right now, we don't do it. There's actually one paper published recently of single-cell analysis of autism versus control brain. And that's very intriguing. We need to scale that up.
Thank you.
Okay, I still see that there are some potential questions. So, but let's take this outside. Let's thank both Shyam and Jonathan for the talks.